I'm sure I did mispronounce that. I'm really sorry. Thank you so much for joining us. I see Sylvester from Kenya. Sylvester from Kenya. Thank you so much for joining us. I see Sarah from Nigeria. Welcome, Sarah. It's great to have you join us. I see Esther Echo from Nigeria. Thank you so much for joining us. I see Deepika from India. Welcome, Deepika. Thank you so much for joining us. Let's keep the introduction coming in while we wait for other persons to join us. I see Olalekan Yusuf from Nigeria. Welcome, Olalekan. Welcome, welcome. I see Mrs. Anita from India. Mrs. Anita, welcome. Thank you so much for joining us. Let's keep the introductions coming in. We keep waiting for others to join us as well. Okay. Let's keep the introductions coming in, guys. There's so much to learn today. Okay, I see Dawood from Pakistan. Welcome, Dawood. It's great to have you join us. Great. We're seeing the diversity once again, like we've been seeing for the past sessions. And it's really interesting to see how much people want to learn about pharmacogenomics. I see Saranya from India. Welcome, Saranya from India. Thank you so much for joining us. It's great to have you here. Okay. Thank you. Let's keep the introductions coming in. All right, let's keep the introduction coming in. For about 3 minutes from the start time, the proper start time of the webinar. Let's keep the introductions coming in so that as much as possible, we'll have very useful time for questions and answers at the end of the session. Welcome everyone. I see new persons joining us now. If you're just joining, just put your name in the chat box and the country from which you're joining us. Would like to see your name and your country, just so we know how diverse the room is. I think that also helps us in our discussions and looking forward. So just put your name in the chat box and the country from which you're joining. I see Sylvia from Nigeria. Welcome, Sylvia. Thank you so much for joining us. Okay, I see Ziphion from Kenya. Hello, Ziphion. Thank you for joining us. I see Dr. D. Rajput from India. I hope I pronounced that correctly. Thank you so much for joining us. Thank you. Let's keep the introductions coming in. Let's keep it coming in. Let's keep the introductions coming in. I'm also going to the group chat to make some noise there for those of us who, of course, are admins. So people know we've started. Okay, I see Siva Guru from India. Welcome, welcome. I see Dr. Esiosa from Nigeria. Welcome, Dr. Esiosa. Thank you so much for joining us. I see Andrew from South Africa. Welcome, Andrew. Welcome, welcome. It's great to have you join us. It's great to have you join us. So it's 4 p.m. on the dot, and our speaker will be joining us in a short while. Okay, I see Baba Hamza from Pakistan. Welcome, Baba Hamza. You've been really punctual. It's really amazing to see that every time you're always part of doing the introduction. Thank you so much for the enthusiasm. Thank you, thank you, thank you. Just joined us. We want to see your name in the chat box, name and your country in the chat box. Thank you. Okay. Okay. If you just joined us, just your name in the chat box. Let's see the country from which you're joining us from, the name and your country. Okay, I think I see Nuhu Prince from Nigeria. I see Winifred from Nigeria. Welcome, welcome. I see Ben from Malaysia. Ben, welcome. It's good to have you join us.
Thank you.
Yeah, okay. You're from Malaysia. Wow.
Yeah, it's already night in my own place, 11 p.m.
Wow, wow, wow.
Yeah.
That means it's been 11 p.m. each time you join us. That's
yes, exactly.
Okay, great.
Thank
Our
you so much.
speaker is in the house. So we're going to move straight now. Welcome, Professor. We're very grateful to have you join us. Thank you so much for honoring our invitation. We're going to move straight up now to the introduction proper and going straight to the webinar. So before now, we've been doing introduction, like persons in the house, stating their names and the country from which they are joining. And there's been so much diversity here. We have a lot of persons here with so much energy and passion to learn about pharmacogenomics. And we're very certain that at the end of this session today, you would certainly get some value from being here. Okay, we would move straight now to introducing our speaker. I'll be reading his bio and then we'll move straight to the presentation so we have enough time for discussion afterwards. Okay, our speaker for today's webinar is a distinguished professor. And before I go over to read his biography, I'd like to state that he's been of great support to this course. And as a matter of fact, he's not only a speaker for today's session, he also doubles as the founder and CEO of one of the collaborating organizations that puts together this amazing webinar series that we're having. So it really shows that Professor Mohammed is actually someone who cares about pharmacogenomics literacy on a global scale, and we're really grateful for that. So I'll move straight to reading his biography now. Our amazing speaker for today, Professor Dr. Mohammed Likwat Raza, is a professor of pharmacology and neuroscience, and as I stated earlier, the founder and CEO of Transcend Science Consulting. We are honored to host Dr. Mohammed, a distinguished healthcare consultant, academician, researcher, and neuroscientist with over 20 years in pharmacology, physiology, and neuroscience. 20 years experience in these fields. His extensive international research experience across European laboratories has equipped him with a diverse skill set in electrophysiology, patch clamp techniques, CRISP, Cas9, AAV production, toxicology, molecular biology, and neuropharmacology. Dr. Mohammed is the founder and CEO of Transcend Science Consulting, where he provides expert guidance in medical research, scientific writing, and pharmaceutical training. Over the years, he has held key academic and leadership positions, including project director at Martin Dow Maka Limited, director at the City Institute of Health Science, and head of department at Ikra University. He has also served as an associate professor at Hamdard University and a visiting professor at the Institute of Business Management in Pakistan. His impressive academic journey includes a PhD in neuroscience from the Charité University of Medicine in Berlin, a postdoctoral fellowship in molecular neurophysiology from Lund University, Sweden, and an MPhil in neuropharmacology from the International Center for Chemical and Biological Science, Karachi, Sindh, Pakistan. He has also conducted research fellowship at University Laval, that's in Quebec, Canada, United Arab Emirates University, UAE, and the National University of Singapore, further strengthening his expertise in neurobiology, pharmacogenomics, and clinical pharmacology. Dr. Mohammed is widely recognized for his contribution to scientific publishing, peer reviewing, and editorial work with high impact medical journals. His commitment to advancing precision medicine, digital health, and AI-driven research has positioned him as a thought leader in the global scientific community. Today, we are privileged to have Dr. Mohammed share his insights on leveraging emerging technologies in pharmacogenomics. I'm very certain that his expertise in cutting-edge innovations such as the CRISPR-Cas9, AI-driven healthcare, and molecular pharmacology will provide us invaluable perspectives on how technology is shaping the future of personalized medicine. So I would like to see the energy in the house now as we welcome our distinguished professor. Thank you so much, Professor Mohammed. The floor is yours now. Thank you.
Thank you so much for the very detailed introduction. I think you have talked a lot about my background and like the experiences I had. So I thank you also a lot of collaborator for this series of pharmacogenomics webinar. So I hope the previous sessions, all the audience have gained a lot of things regarding this important topic. So a little bit I want to talk about that, and since this is a very you can say a very hot topic these days, so I'm gonna touch upon little bit different technologies. Might be if any one of you haven't heard before, so there will be some technical terminologies. There might be some something which is like new for you, because it's a growing technology, as well as people are using nowadays, especially in the drug discovery, especially to target the new disease therapies for utilizing the various pharmacogenomic approaches. So let me share my screen first. All right. So can you see my screen?
No, sir.
Okay. Sometimes it may be a problem. Okay. Let me. Okay. Just give me a minute. I can just quickly relog in so that it might be because of that. Give me a minute, please. Okay. So now I think the option for sharing screen is disabled now. So cannot share the screen right now. It was appearing before, but right now it is not appearing. Okay, yes, it is. Fine. All right, so can you see the screen now? Can we
Yes, sir.
confirm me if yes?
Yes,
Okay,
sir.
Yes, sir. It is appear now.
okay, great, super, super. All right, so first of all, a very brief introduction what the pharmacogenomic is. If you see the word itself, it is self-explanatory because it's a combination of pharma, which the people from the medical field easily know. The pharma means like drugs. When we talk about genomics, it means something related with the genes. So when we combine the drug with the study of the genes, so it become a pharmacogenomics. So in journal, when you see the pharmacology definition, the definition of pharmacology means you study the drugs, the drug effects, the drug properties, how it interact with the body, how it interact with different receptor site and produces therapeutic action. So in journal, if you want to see the effect of drug, not only on the body, but at the level of genes, how the response has been changed. So the pharmacogenomics provide you a platform in order to study the effect of the drug at the gene level. And this field is actually not very old, since if you go see the history of the genomics field, it's nearly, you can say 35, 40 years before. So back in 70s, the starting of the genetics starts, and then afterwards a little bit of the refinement of the genomic phenomena, early 80s. So you will have certain techniques in which you can study the genetic property utilizing the various approaches. And if you see these approaches at the level of drug effects, so it become a pharmacogenomics. So the field is not very old. It's a very new field. As I said, like back in 70s and 80s, you will have these effects. So in order to use the various genomic techniques. So in journal, when we combine these two things, as I said, like the drug effect as well as the study of the genes, so these become the pharmacogenomics. So what is the purpose of studying the drug at the level of genes? Why we are like more focusing on that? Why we need to apply these genetic approaches in order to find the effect of the drug? What is the logic of that? What is the objectivity? What is the purpose? So very simple, because we know there are because of the emergence of the field of genetics, we know there are a lot of diseases, or you can say the most of the diseases have some certain genetic links. So there are certain diseases which are like genetic diseases using the chromosomal defect at the X chromosome or the Y chromosome. But now we know, like at the level of the CYP enzyme, which is like one of the key factor for the drug metabolizing in the liver. So you have the cytochrome P450 system, and they have like more than 232 enzymes in that. So if these enzymes are basically homogeneous, heterogeneous, or there is a phenotypic and if we see the phenotypic properties, if we see the physical property of these enzymes, they might get mutated, they might get changed. How they are changing? When they are changed, they can definitely affect the properties of the drug. So this is a very key player here when we know about the drug effects. So we know drug goes absorb. First, there are 4 phases of the drug. When it goes into the body, it absorbs, it metabolize, it distribute, and it eliminates. So when the drug is metabolized, it's basically because of the enzyme system which is present in the liver mostly. So the majority of the drug gets metabolized in the liver. So if the liver enzyme system is basically have certain defect, and that these defect is basically if there is a change in their genetic setup, there is a genetic disposition in that. So you have to study these genetic dispositions so that you can address this issue, and then you can easily address the effect of the drug. Because if the drug is not metabolized, or the drug is metabolized very quickly, or the drug is metabolized very lately, so it can eventually turn into either a toxicological effect, or the intended effect is not be achievable, because if the drug is metabolizing too fast from the body, so that you will not be able to get the intended therapeutic action. So we know if we apply certain techniques in that, and then we can come up with this technology, how we can study the property of a various enzymatic system which is present in the liver. Utilizing the genomic technique, we can come up with the right solution for the drug which are early metabolizer, or which are like the slow metabolizer. So this is the purpose, like we can easily address a lot of diseases which are due to the genetic disposition in the either in the enzymatic system, either at the receptor side. Because we know there are a lot of diseases which are now purely have a clear-cut genetic linkage. For example, let's talk about if we take the example of the epilepsy. Epilepsy has more than 100 mutation in the different receptor site. For example, there is a mutation in the sodium channel, there is a mutation in the potassium channel, there is a mutation in the SK channels, there is a potassium in the calcium channel. So if we know these mutations, we can easily address the properties of the drug. Because if these channel are for example, if the drug goes on the sodium channel, and the sodium channel is basically mutated due to any genetic changes in that, so the drug will not going to work. So if we modify the drug, or if we modify the receptor dynamics utilizing these approaches, so that means we can provide the patient with the relief, as compared to in a situation when we don't have any clear-cut resolution for these conditions. For example, 35% of the patient are not responding to the drug, anti-epileptic drugs. So what is the solution for these patients? Because if you say there are 80 million people nowadays in epilepsy throughout the world, and out of them 35% are not responding to the drug. So how can you treat such patient? And it's a huge number actually. Likewise, other diseases we talk about, like Alzheimer's, we have a clear-cut synaptopathies in the Parkinson's disease. We have the clear-cut tauopathies into the Alzheimer's disease. We have clear-cut lots of meninges, as well as oligodendritic cell changes in the multiple sclerosis. Same with the ALS, same with the Huntington disease. How can you treat these patients when we know there is a clear-cut genetic disposition in these patients, either at the receptor side, or there is a different part of the brain actually? So we can actually modify these things utilizing the genomic approaches. So the objectivity is you can have a very selective and optimized drug, as well as not only the drug, but you can also optimize the dosing. Because if for example, if the drug is working and the steady state of any drug is like for 6 hours or 8 hours, that means we have to repeat the second dose after 8 hours. So what happens if there is any change in the system, in the metabolic system, or in the liver, or in any receptor side? This steady state is not maintained because of various factors. So because this is again, there are a lot of factors that are involved in these that can contribute either in the delay of the distribution of the drug, or either the receptor binding get affected due to to any of the reasons. So we have to add these points. So in order to rightly pick up the dosing phenomena and the dosing frequency of the drug, so we can easily address these points through the pharmacogenomics. Similarly, we can, as we know, there is a toxicological property of the drug. So utilizing the pharmacogenomic approaches, because if the drug is agonist drug, it might goes to a various receptor site. For example, let's take an example of a serotonin receptor. Serotonin has a various subtype, for example, 5-HT1, 1A, 1B, 1C, 5-HT2, 2A, 2B, 5-HT3 receptors, and 3 receptors further subdivided into 7 subtypes. So, you know, there are huge variation, the classes of the receptor as a subtypes actually. So when a drug goes in one receptor, it can also go to the other subtype of the receptor, and that can eventually produce their side effects. So how can you maintain these side effects? How can you manage these side effects if you provide specific exclusive approaches that can only supply the drug to a targeted specific subtype of the receptor, so that you can avoid the toxicological or the ADR of the drug. So this is another big application of the pharmacodynamic, in which you can easily target the drug toward the specific subtype of the receptor, and the drug goes into a specific receptor site and produces effect. Third important factor that can the benefit of the pharmacogenomic study is basically the improved and improvised basically efficacy of our treatment overall. So we can optimize the treatment dosing, and then the overall treatment modality can be improved if you have the clear-cut efficacy data, if you have a very sharp knowledge about their receptor dynamics, so we can actually improve the efficacy of that drug. So this is the key fundamental objective of doing a pharmacogenomic study of any drug or of any specific disease condition. So these the main and core can say the objective of studying the pharmacogenomics. All right, so what are the challenges? You know, every technology has some limitation or some obstacle as well. So how can you, because, you know, as the technology comes, it has a lot of things to deal with that. So still, as I said, it's not a very old technology. So you have a lot of still challenges that you have to take care of that. For example, I'm going to touch about a very superficial one, not all the key players in the obstacle way. So first of all, you know, as we say, so if we talk about the phenomena of the drug interaction, so we know a lot of drug interact in between each other, or there is a food drug effect. So you can have the food drug interaction, you have drug interaction, you have pharmaceutical drug interaction, you have pharmacological drug interaction, you have physicochemical drug interaction. But now when we talk about the working of the drug at the level of gene, so there is a little data available that can actually give you an insight about the gene drug interaction. So DDI is very common, food drug interaction is very common, pharmaceutical interaction is very common. But regarding the data we have available so far at the level we are.
I'm so sorry to cut in. I'm really sorry. I don't know. I'm so sorry to say, but we have persons in the house who are not really from completely English-speaking countries, so I think they might be having challenges.
Okay.
Running with you, yes. I think if you could slow down a bit, it would really help.
Okay, no problem,
Yeah.
no problem.
Thank you so much.
No,
Thank you so
you're welcome. No problem. All right. So, you know, since this field is very new, so still we have to go a lot about these interaction with the genes. So the data available, if you see in context of, you know, there are more than 4,000 drugs available we have so far to treat various conditions. So we don't have the enough data that on the basis of that we can say this condition or this drug is effective, because we have all the data available for the gene drug interaction. So this is the missing component, and this is one of the obstacles that we have to understand the working of the drug with, because, you know, we have a lot of gene in the body, and the drug is actually it can, as we said, like it can interfere with the genes sometimes. So we don't have the enough literature right now, the enough studies that can actually correlate the gene drug interaction. So mark this point, this is another special field. Like the drug interaction, we have the gene drug interaction. This is one of the obstacles. Number two, as we said, like since the studies is still scarce in this field, so definitely we will have the second phase of any any studies, first phase is basically on the animal, and the second phase is basically on the patient. That is a clinical phase. So likewise, when we have a very limited data available for the gene drug interaction, so definitely we have a limited clinical application or validation of these drug gene interactions. So we are not fully aware whether these genes are, when they are like affected because of any situation, either it's a disease state, it's an environmental factor, whether it's stress, whether it's organic cause, whatever situation is that can interfere with the genes. So when the drug goes with such certain genes which are already undergoing any abnormal change, so whether these changes in the gene and the interaction with the drug can be clinically validated. So this is one of the second limitation, that we don't have the enough clinical validation of the such gene and drug the interaction. Number three, basically, when the techniques comes, it's very costly. So doing the pharmacogenomic studies is not easy task for every lab, because it involves a lot of specific chemical, a lot of enzyme system. For example, RNA-seq is one kit for RNA-seq costs you 2,000 might be, depending on what kind of RNA-seq you're going to use. So you can imagine if you're going to work on an experiment that require like 50 kits. So 50 kits of RNA-seq, so multiply the 50 kits of RNA-seq, multiply by $2,000, so it become like nearly $100,000 for just one set of experiment if you want to focus on very precise specific genes. So this is not easy to bear the cost of high cost of pharmacogenomics studies by every lab. So again, like this is a very superficial points that I'm telling. Still, there are certain things which are very cost effective, so which are not that much costly, but I'm talking in general, in total actually. So this genetic testing involve a lot of complications sometimes, along with the money factors, along with the technology issues, along with the equipments that you need for that. The fourth point and fourth obstacle in this field is basically, so when you come up with anything new, it takes time to get integrated into the existing system. So, we have a healthcare system in every country, every region. So Europe has their own standard, the Americans has their own standard, Asian countries sometimes have their own regulation, and authorities has their own guidelines to adopt anything. So healthcare integration would be another challenge if you talk about to emerge and to integrate the pharmacogenomic approaches into the existing healthcare system. So this is a fourth obstacle that we can say that is an integration into the existing healthcare system, because when you integrate something, it also affects the workflow, because for example, if you're going to implement AI in your diagnosis in your hospital, so what is the current situation in your hospital, current methodology which are you using nowadays? If you integrate something new in that, so it will require time, and it can affect the workflow as well. This is considered as one of the obstacles there. So still, when there are some obstacles, there are still some hope, so which we call it like the improvement phenomena. So how can we improve these things? Just because if you as you said, like we're still unaware of a lot of parameters of the gene drug interaction. So what we can do, we can start developing the standardization of these testing and reporting actually. So this is actually with a mutual collaboration between the university, academia, and hospital and the regulator. So unless all these components will not come in to close collaboration, it will not be possible to standardize and to report it. For example, if a big hospital has a huge budget and they are focusing on some pharmacogenomic approaches into their research and development projects, but what about the lower hospital which are lacking the huge budget? What are like small clinics? So there should be at least a reporting system which should be centralized one, so that they can gather the information from small to a big tertiary care hospital and the industry which are working on the genomic approaches. So standardization is one of the key that we can improve so quickly anything that we want to implement. Second thing is, like, of course, when you come up with something new, you have to educate and create the awareness. Like the purpose of this seminar from this organization is to make people aware of this important technology, this important emerging field of pharmacogenomics. So that's why they have arranged this series of pharmacogenomics seminar. So the purpose when you get people aware of the anything new, or you educate them, there is a chance of high acceptance. So in order to implement something, if there is a resistance or there is a rejection from the authorities or from the public, so it will be very difficult you to implement anything. That's why if you want to implement anything in future, so you have to educate for that. You have to make aware people of this importance so that they can easily accept when you're going to implement that. Third thing, as I said, like, since this is a healthcare sector, and healthcare, there's strict regulation and bindings, they have a strict laws and regulations. So from the local authorities, from the government, from the Ministry of Health, or from other regulatory bodies that are actually part of a healthcare system. So this is another important point, that to sit together and to devise some policies, some regulation that can flourish this emerging field of pharmacogenomics, so that when it should be implemented at any stage of in the coming future, and in their city, in their country, or in their hospital, so there shouldn't be any ambiguity, or there shouldn't be any confusion of why we are implementing it, what would be the consequences of that. So there should be a clear regulatory framework of that, how you're going to test these pharmacogenomic approaches into a real patient time. So this is the areas that you can improve actually and overcome your the obstacles. All right, so we're going to touch upon a little bit one by one some of the tools, some of the techniques, and some of the emerging technologies which is part of the pharmacogenomics. So one is very common one, because this is also used in genetics very commonly, which is NGS, that is called next generation sequencing. This is a technique. And what is the benefit of this technique? The benefit is that you can produce the small part of if you're going to perform an NGS, so you have a small you can say the sample size. If you have a small testing material, that you can produce a lot of things using the high throughput screening. Actually, again, the high throughput screening is another technique that you integrate in that, and you can multiply for example 10 item into 1,000, and from 1,000 you can do a 10,000, and from that you can cross like the million. So this is the advantage of that with a small if you have a small gene pair, if you have a small RNA part or DNA part, so you can replicate that and perform a lot of things utilizing these things. And second advantage of doing the NGS is basically, if you consider an example of a long peptide, which has like let's suppose 2,000 amino acid in that. So for example, if there is a slight change in the amino acid sequence at the base pair level of 2,000, because a long peptide 2,000 amino acid, and if there is a slight change in, let's suppose, at the base pair, which is at the point of 159 or 160. So there are techniques which cannot detect slight variation. So the beauty of the NGS, it can detect a very rare and a very small amount of variation into the base pair. But this is the beauty of the NGS that you can do. And not only this, it can provide a very comprehensive and a dynamic genomic profiling. For example, if you want to do the study on the effect of a drug into the patient with the Parkinson's disease, and you want, for example, we know there is a problem in the dopaminergic neuron when we talk about the Parkinson's disease. So what actually at the level of genomes, there is a changes or alteration that can causes this disruption into the dopaminergic circuitry or dopaminergic neuron, or there is also abnormal signaling of the cholinergic pathway, because these are two major things which are impaired into the Parkinson's disease. So the cholinergic pathway as well as the dopaminergic pathway. So what is the correlation of these abnormal signaling into the brain in context with the genes which are regulating the dopaminergic neuron and which are actually controlling the dopaminergic neurons and as well as the cholinergic neurons? So if we know these genomic profiling in depth and in detail, so then we can come up with the right solution for the Parkinson's disease. Like with the same, if you apply the same thing on the Alzheimer's disease, these tau protein, or there is a protein misfolding into the various region of the brain, because these aggregation of the alpha beta tau proteins that can stick with the neurons in the brain, and then you feel a severe category of the Alzheimer's disease. So how these actually protein they gather, they aggregate. So if you know the genomic phenomena and genomic profiling, so we can come up with the right solution of the treating the Alzheimer's disease. So this is the beauty of the NGS. It can provide a very comprehensive genomic profiling. So these advantages become technically the application. So application is you can perform the WGS. What is WGS? This is called the whole genome sequencing. So if you want, for example, now you can have the whole complete genome of your cell. Although it is a little bit expensive, if you want to explore your complete genomic sequence, that's possible. So you can do the WGS, which is a whole genome sequencing. So this is the one which is like you can do. And then when we talk about a little bit, if you want to do something with the disease, to develop some therapies, so we have another type of sequencing which is called the exome sequencing. This is very common in, especially in the neurodegenerative diseases, because all the vesicles in the neurons, for example, whether it's a dopaminergic vesicle, if it's a cholinergic vesicle, or it is a GABAergic vesicle or circulatory vesicle. So all these vesicles, when they come to synaptic button, when there is a synaptic release from these vesicles, the neurotransmitter is shed into the synaptic cleft. So all these neurodegenerative diseases have interference of these release phenomena, either or reuptake phenomena, or there is basically the exomic role in that. So we can do the exomic sequencing and find out what abnormality and irregularities are existing in there, so that if we found these irregularities in the exomic level, so we can control the next step, and then we can prevent these diseases actually. And then we have another thing which is called something which is more with the patient oriented, which is called the targeted gene panel, in which you do not focus on one gene. Rather than you focus on the cluster of gene. For example, let's take the example of the schizophrenia. So we know there is a mutation in the chromosome number 3, chromosome number 7, chromosome number 11, and some studies said even chromosome number 16 and 17. So if you want to target like the panel of gene, that means not one gene, but a multiple gene or cluster of gene, then you apply the another technique, which is basically a targeted gene panel. Likewise, another beauty of these NGS is you can easily do a very personalized study on the tumors actually. So whether the tumor is basically benign, tumor is basically spreading, tumor is metastasized at the level of stage 1 or 2 or 3. So you can do the whole tumor. There is a tumorigenic phenomena which is existing into the body. You can find out these tumor biomarkers, and then you perform a personalized cancer treatment. So this is one of a lot of pharmaceutical industry are working on this tumor profiling for the precision medicine development for the cancer. Various kind of the solid tumors are being treated, will be treated in the coming future, utilizing this NGS based personalized cancer treatment actually. So if you take the example in terms of cost, for example, as of 2024, so you can do a sequencing actually under $200, and when it was actually back in 80s, it cost one sequence to do like $40,000. So it's now come to because of the various kind of chips available. Kits available. So on the basis of kits, you can get the maximum WH sequence of anything. So which is like a little bit cheaper compared to if you go like 20 years back when it was $40,000 for a sequence section. Imagine $40,000. So now it's like $200. So this is the emergence of technology. Now it has been reduced to a significant level that can be adopted by many countries and many labs that can afford that thing. All right, there is another technique which is called RLST. That means basically long read sequencing technologies. So you don't need to like digest all these in one lecture. Just I'm telling and the main technologies which are part of the pharmacogenomics. So why when we use these long read sequencing technologies, for example, if you have a very robust kind of mutation diseases in which you have the sequence of a base pair is not only linear but also branched. So in order to find the variation in, for example, let's take an example of any abnormal cancer. The cancer is spread uncontrollable and the different side. For example, from one part of the body it can go to the other part of the body. So this spread is very, have a huge variation sometimes. So in order to find out the spread cancer, so if you want to do a change, if you want to see the change effect into the different base pair within that, for example, if a lung cancer is spread from one part of the lung to the next, another, the right lung to the left lung, or from lung to it goes to the stomach, or from other parts. So this variation actually is because the lungs cell is basically elastic cell, and the cell which are present in the stomach are different than the lung cell. It's not elastic that much as the lung cell are. The lung cell has the highest capability of the elasticity. Also, although the cells which are present in the stomach also has elasticity. So because but overall the properties of the elasticity and the power of the elasticity is different than the lungs it has. So if you want to study these variation and structure variation, so you can use this long read sequencing technology. So the first one. The second one, it provide you a very clear cut data if you have a very complex rearrangement. As I said, like if you see the cancer dynamics of the cancer cell or the tumor cell, which is grown so much, so it is a very complex rearrangement of the cell altogether. Some cell are like of a different, cytoskeleton of that tumor cell or the cancer cell has a huge variation in that. So you can do the complex rearrangement and then perform these long read sequencing. Third is basically haplotype phasing is another type of when you study the basis of genetics. So you have the haplotype phasing. So it can provide a very fast or enhanced haplotype phasing, and this help actually to regrow the cell and to study the cell in a quick fashion. So these are the advantages of the long read sequencing technologies. So when we talk about the impact, if we perform these long read sequencing technology, what will the potential impact in our field or in our practice? So when we say, like, when we characterize very precisely with the structural change and complex rearrangement of any disease, tumor, cancer, so that means we can have a very clear-cut demarcation. So we can, on the basis of that clear-cut picture, either we can predict like whether we need to remove that thing precisely because we have a characteristic of that abnormal thing, so that we can perform for example a resection of that part so that we can remove that cancer or oncogenic part, or if there is something which we can treat through certain gene therapy based approaches. So now the purpose of getting this complex rearrangement and complex variation, then we can also apply the gene therapy. So nowadays a lot of groups are working in various diseases to treat these diseases through the gene therapy approaches. So this is one of the very potential impact. If we know the variation, the structure variation, we know the complex rearrangement, then we can perform the right gene therapy using a CRISPR. So as introduction, he was mentioning of the CRISPR technique. So with the CRISPR technique, we can load into the adenoretroviral specific sequence, a specific promoter of the gene. That promoter can take a lot of different promoters, and if there are 4 or 5 variations, so we can chip into this adenovector 4 or 5 different promoters. It can go there and it can actually make a new base pair and it can heal that thing. So this is the way actually when we in the coming future, when we see the lot of diseases will be treated through the gene therapy using CRISPR or other techniques actually. So this is basically and just to let you know one example here. Just to know an example here. So there is like PacBio, which is a sequencing used for a variation into there is a there are if we talk about the liver enzyme system, there are more than 230 enzyme system there, isozymes there. So one of the key isozyme is basically CYP2D6. So you can see the majority of drug has been metabolized by CYP2D. So there is a specific sequencing tool that can actually use to predict or to find out, to investigate if there is a variation into the CYP2D6. So that's called the PacBio's Hi5 sequencing tool that you can use and find out any variation or any abnormality in the CYP2D6 isozyme actually. Now, the easy technique, which is very common in the various lab nowadays, these labs are using basically single cell sequencing. For that, as I said, like in the start, I said you can use the RNA-seq. Basically, it's a whole kit that you can order from a company, and you can actually apply these kit at the level of the RNA, at the level of DNA, or if you want to see a response to a drug, then you use the basically STAC. So these are the kits available that you can use in your lab, and then you can find out various kind of you can detect even the rare diseases. You can detect the effect of the drug. You can detect basically the tumorigenicity of a particular type of tumor. So this will help actually to develop a very targeted and a cell specific drug therapy. So as I said, like as the name is indicate, like single cell sequencing, that means basically you're going to analyze a single specific type of cell, and because this is in the initial phase easier, because you can see the effect of the drug on a specific single cell. So or a specific type of cell. For example, if you want to check the mutation which is in the glioblastoma cells into the brain, so what you can do, you can use a specific basically cell sequencing, and then it can goes directly to the if the patient is a glioblastoma. So these are the things we can develop. For example, if you want to see the tumor necrosis factor, if there is present or not, the tumor growth factor, if you want to study that, if you want to see the BDNF actually, and if you want to see any specific peptides there which are responsible for these things, so you can focus on a specific peptide there, and then these sequencing will go a specific peptide, and then they can mimic their action, and then they can confirm whether this is present or not. And if they are present, because we know if the tumor is there, that means basically there is a specific cell biomarker. Yes, someone mic is open, I guess. Okay, so so these single cell sequencing techniques are very common in the lab. So you can identify a lot of things with a single cell drug response. You can point out basically the rare population of the cell with the efficacy and toxicity of the drug. You can study in a detail the tumor property actually. So whether there is a heterogeneity or the homogeneity, and then on the basis of that you can develop a very cell specific treatment. So this is one of the very potential one, and I said like it's very common nowadays in the labs. Most of the lab are using these RNA-seq. So for example, if you want to identify the specific drug targets, so you can use the RNA-seq. If you want to detect genetic variants, then you can use the basically DNA-seq. So if you want to like perform a specific study of the drug response, then you can do the ATAC-seq. So these are the some of the important techniques that you can apply, and then it's very easy because you have prepared a kit from the company you can order, and then you can follow all the steps which they have sent along with the kit, like what steps you have to follow, and then you can easily find out any sequencing differences or mutation or the changes of the variation into the specific scenario that you want to focus for that. All right, so and then the technology emerged. So this is another one which is better than the RNA-seq, which is called basically the rapid SNP detection technique. SNP basically single nucleotide. This is a single nucleotide. Remember, so in a research, always you should know your aim. So if you want to study the single cell, you use the RNA-seq. If you want to study basically the nucleotide, then you will perform the SNP technique, which is single nucleotide techniques that you use. So nowadays, if you compare like 10 or 15 years back, you have a very traditional one. Now you have a very you can say the automized kit. So just utilize this awesome automized kit, you can perform easily this rapid SNP techniques there, and you can detect actually various things actually. Not only this, for example, there is a very common one which is called a SMART-M2. With the SMART-M2, you can amplify the DNA For example, those who have performed ever this, the technique of the RT-PCR, the purpose of the RT-PCR is the same, that you can amplify a specific protein or amino acid sequence. So in the SNP technique, again, we have with this kit of a smart M2, you can amplify the actually DNA sequences. And when you amplify the DNA sequences, you can study a lot of things. The purpose of this single nucleotide, the polymorphism, is basically to detect any change at the level of the nucleotide. So this is the difference. We are not talking about the cell, rather than we are talking about the nucleotide sequence. So there is this is exclusive technique when you want to do any change, any variation into the nucleotide peptide. So then you perform this one. And previously it was a little bit tedious. It might take like 48 hours. Now you can get the result. You can imagine how the technology is getting faster and faster. So with the rapid SNP kits, you can do it like within 45 minutes actually. The life is very easy. Like if you do a PhD like 10 years back, for these things you have to wait like sometime 2-3 days. And sometime if you miss any step or did something wrong in the step, so the whole exercise goes wrong. So now it's all automated, it's all perfect. So what the beauty is, you can simply perform these kit because when when you get these kits, you have basically the whole step by step procedure, right? For example, which reagent you have to mix like together, how much waiting time you have to give. You have to do, for example, you have to centrifuge it like 7 minutes at like 5 degrees or 20 degrees. Everything is written there. So you have to just follow step by step, and you will at the end of 40 minutes, 45 minutes, you will get your results. Amazing. So this is the beauty of this thing. So what is the benefit of these rapid SNP techniques? Basically is, as I said, you can detect the mutations at the nucleotide level. So all the diseases of genetic diseases, if there is a variation, if there is a mutation in the nucleotide peptide, or if there is any abnormality, irregularity in the nucleotide peptide, you can easily detect, and that can provide a very potential help to develop a patient care on the basis of these genes. Yes, if we compare it with the traditional, as I said, the genotyping methods, so this is cost effective because it's a single kit that you can apply with the small reagents, and then it's all done. But if the local and the traditional genotyping methods are not only tedious and longer, but also it involves so many things that it costs a lot as compared to these kits now. So, and then one of the beauty, you doesn't need a lot of different other 2 or 3 equipments. Rather than you can perform it in 1 or 2, maximum two equipments. So this is another thing that is like very important. So for example, if you want to, just for an example, if you want to see the variation to develop antiplatelet therapy, because the antiplatelet drug are basically metabolized by the CYP2C19 enzyme. So if there is a variation in the CYP2C19 isozyme of the liver cytochrome P450 system, so the antiplatelet drug are not gonna work. So the heart patient, the patient who are with the coronary syndrome, so if antiplatelet therapy doesn't work, they might have a life-challenging situation. So you can detect why these drugs are not working in these patient or such patient, then you can easily perform rapid SNP detection method, and you can detect whether there is a defect in the CYP2C19 or not. What you can do if there is a defect, we know like there is no so far therapy that we can correct this defect, but we can hope. But now what you can do, you can use alternative treatment plan for such patient, because antiplatelet therapy will not going to work in these patients. So what you will do, you will choose a drug that will not metabolize by CYP2C19 system. Now, which is everybody knows, is basically artificial intelligence. Everywhere you see AI. So again, the pharmacogenomics technique has been coupled with the bioinformatics as well as artificial intelligence. Again, the bioinformatics is a totally different field, but now nowadays we have all the thing multidisciplinary. So what we do basically, we combine the informatics techniques and artificial intelligence so that we can predict the future. For example, imagine you have like 32,000 base pair. 32 is enough, is too much. So to physically perform these things, it will take a lot of time, might be a couple of years. So with the AI tool, what you can do, you can develop algorithm, you can train the system, and then you can predict, like if I load a base pair of like 50,000, and then we can predict like if there is any variation in the sequences, if there is a change in the sequences, and if it is there, then at which point it is there. So instead of like applying a lab technique and to find out these base pair variation and at which phase, at which point, then it will take a lot of time. So we can do it with the integration of the artificial intelligence. There are so many tools which are available, but like couple of them I can quote you here, like deep variant. So if you want to detect a variation into the sequencing data, so the deep variant is a tool that can actually help you find the accurate variation. Let's suppose you found out a genomic sequence of any patient, just load the data in the deep variant, and it can read and give you the result of variation within second actually. Likewise, if you want to find out any drug gene variations, is any association, if there is any gene drug interaction, there is basically I must mention here, still these are not like well you can say the well effective, because still the field is growing and we need a lot of improvement, but at least it will help you to get somewhere in a very fast fashion. So if you want to use a tool which is called the PharmGBK, so it will give you basically about the gene drug responses, any changes in that. So if you want to analyze your overall genomic results, so that there is an AI assisted tool, just like in the AI diagnostic, we use the CDSS, like computerized decision support system. We have also in the pharmacogenomics, this is called CPIC guidelines, that are actually analyze your data and compare it with the CPIC guideline, and then say, okay, you are violating this point, it is not in compatibility or in alliance with the guidelines, so that you have to review results. So this is, you can say, a sort of a cross check of your final results. And then we have a big drug bank AI, in which you can find out the drug target interactions actually with their side effects. So these are the integration of the AI in the field, and it's a huge field actually. A separate, you can say several hours topics is needed to actually detail explanation of the AI integration into the pharmacogenomics field. And then when we talk about the application of the bioinformatics, again, this is a huge field. So just to give you a glimpse of that, so it's all in a nutshell. So integrating AI with the pharmacogenomic tool, you will give a very large scale data analysis in a short period of time with the accuracy actually. So when we talk about which we have discussed in one of the obstacles, so integration into the healthcare system basically. So it's still there are countries, developed countries, they have already chipped in. In USA, there are various hospitals have already have the pharmacogenomic chipped in tool where they are using and they are applying the personalized therapy. Actually, the purpose of the pharmacogenomics basically to have the one-to-one personalized, or you can say the tailor-made medicines actually, in which the specific medicine should fit for that person. It's not like the acetaminophen should be fit for all population. No, whether it is fit in this patient or not, so pharmacogenomic will tell, because when we have if the N-acetyl enzyme, it has an N-acetyl moiety in that, so it is specifically metabolized by N-acetylase enzyme. If in some of the people it is absent, so if it is absent, it will not going to work. So likewise, other drug. So if you imagine this is simply a acetaminophen, which is just an analgesic drug. What about if you're taking the antihypertensive drug, anti-diabetic drug, or anti-hypercholesteremic drug? So these drugs actually sometimes can make a life worse if you are not responding to medication. And pharmacogenomics will tell because the enzyme system is not present or it's not working well. So you have to actually change to other drug. So in these countries, some developed countries already adopted these things. So They have integrated into the system with their hospital main, in all the developed nations, they have a centralized software system for diagnosis, for the lab report that integrate everything all together. So they have pull up this data with which is called the electronic health record with that of the pharmacogenomic tool, and then they can get in a quick fashion of these patient analysis with their lab report, with their diagnosis, with their pharmacogenomic pattern, and then they can suggest like this work or not actually in this patient. So this is basically the integration in the developed country. And if we talk about in our country, we have to actually work a lot on that. Otherwise, we cannot provide a very specific and personalized medicines to our patients. This is the Ignite is one of the very big network for the pharmacogenomic and the testing and the clinical decision support system. So if you the big hospital are using the Ignite network system because it integrate the clinical data, the lab report, and then give you in a single screen the decision. This, for example, if the prescription is like 7 medicine, he's like diabetic, he's hypertensive, he's asthmatic, he's like also suffering from arthritis. So 8 to 9 medication were on the prescription. So this support system will actually give you the most appropriate, accurate analysis, and then on the basis of pharmacogenomic pattern as well as the AI integrated tool, and it can tell, okay, out of these 8 medication, 6 will work and 2 will not work because you have to replace this medication as a patient has the variation in the CYP enzyme system. So you have to choose something which can metabolize, but that's enzyme system which is present in the patient actually. So these are basically integration impacts if you integrate the pharmacogenomic into our hospital system, into an overall healthcare system. But this need actually a centralized approach actually. If the data is not centralized, it will not going to work, because centralized means all the lab reports, all the imaging report, the radiology, everything should be at one place, and then these pharmacogenomic the pattern of the pharmacogenomic sequence of the patient should be also stored there, so that it can analyze the sequence and then come up with the right result for you in order to provide a better healthcare to that specific patient. The thing is, when you have these things, again, this is one of the challenges basically to maintain the privacy and the security of that huge data. So always this is one of the problems. So that's why the company put the firewalls and take the strong and the firewalls and take the basically expertise of the IT people, so that they can safeguard the data, because the data privacy is very key element in the West especially, and not only West but all, for the hackers also, this is like a gold, so they can sell the data at the dark web. So this is another feature that you should keep in mind if you're going to implement. Not only implement, but you have to provide all the measures that can safeguard the data and the privacy of your patient. Otherwise, if it is being breached and hacked, so your organization will be actually target of a lot of critics, because if you implement, then you have to safeguard the privacy and the confidentiality as well as the security of the data of the patients. All right, there are a lot of solutions for that, but again, the developing the CDSs again need expertise, technical team that can be comprised of IT people, AI professional, and healthcare professional. All together, we can come up with a very sophisticated and idealistic the pharmacogenomic CDSS system. So future direction, the generally. As everybody can see, the AI is taking the lead everywhere. So if we need something very targeted, very focused, we have to adopt basically WGS in combination with that of the AI. And when we talk about the omics technologies, the omics is just not like pharmacogenomics. It's like if you want to see the transcription error, so you have to do the transcriptomics. If you want to specifically study the protein, then you have proteomics. If you want to study, for example, the metabolic system, then you have the metagenomics. And you have if you want to see the changes into the various pathways that are the pathways of the energy, like TCA, HMP shunt, glycolysis. So you have the glyomics actually. So, it's not just one thing. You have like specialties in that. In those who are interested, then you can dig out information for that. So it's one of the emerging field that can not only provide therapy for the general disease, but it can also be provide a window for rare diseases. And not only rare disease, but also it can provide something which we can we can be hopeful for, such as diabetes, such as the arthritis, such as other neurodegenerative diseases, in which we can apply the gene targeted specific tools, and that can go into the specific part of the brain, or that system, or pancreas, and then that can mimic the right physiological supply there, and then you can treat these diseases with the help of these focused gene mediated tools actually. So in conclusion, altogether, we need a collaboration between a researcher, clinician, and industry, and then we need the huge investment into the academia so that we can perform these kind of researches to come up with the ideas. Before that, we need with a good lab and infrastructure. Otherwise, we cannot perform these high-tech equipments without the high-tech equipment, these techniques. And then again, we need a strong actual advocacy, because we need the regulation and policy, ethical standards, and then along with that, we should have like how much data we have to share with which stakeholders. So that means we need a clear guideline for that from the regulators and authorities and the government, so that we can get the right benefit of this technology. With this, I stop here, and if you have questions, please go ahead.
Fantastic. Thank you so much, Professor. The session was as informative and very exhaustive as possible. Thank you so much for the very, very useful insights. So we will move straight to taking questions now. We're going to be doing it very orderly, just like we've been doing in the past sessions. Okay, I see Professor Nancy. I'm going to, you can unmute your mic now, Professor Nancy, and ask your question, please.
Yeah, hi. Thank you, Destiny, and thank you for your speaker for this very comprehensive lecture. I was just wondering if the speaker can give us an insight on how we should proceed in low and middle income countries. Can he advise us of what kind of testing is better in a clinical setting, in terms of money, in terms of reproducibility, accuracy, etc. And another question about the rapid SNP detection, because you said that this can be used in point-of-care testing. So how many SNPs can be detected in one go in this type of point-of-care testing? Do you know any of these that are present and they can be utilized in these days? Because I know that they are very strong on these point-of-care testing in certain clinical settings. So do you know if there are commercially available kits or commercially available, let's say, detection kits or something that we can buy and use in our countries? And thank you very much.
All right. Thank you for the two questions. So first of all, like the first part, like in the low income countries, what should be the step to move forward? So, first of all, in the lower middle income countries or the country with the less budget, so one of the important thing, for example, I can mention you about, there is international research network, which is called Southeast Asian Pharmacogenomic Research Network. So what you should do is basically align with these kind of organizations. So they supply a little bit, a training, as well as a small budget to carry on some of the researches. For example, as I said, this organization, which is called SIAPharm, the Southeast Asian Pharmacogenomic Research Network. So if you are focusing on any disease burden for malaria, HIV, tuberculosis, and non-communicable diseases, so they provide you with a lot of guidelines. They have the structuring. They can supply you some of the resources that you can utilize their resources, and they fund you not huge money, but at least to you can perform in your lab actually. One. Number two, what you can do, because the pharmaceutical industry are richer than the academy actually. So what you can do, you can collaborate with that's why I always said the industrial and academic liaison is very important to flourish. So likewise, if we have in any country which are having some of the big pharmaceutical there, so you can collaborate with them, you can liaison with them, so that because they can provide you fund for that to execute these drugs, to these studies and techniques. So this is point number 2. Point number 2, basically, itself, if you there are certain big international grants, so you can, if you're from the any underdeveloped country, so you can apply. For example, Bill Gates and Melinda Foundation, you have like even the Microsoft, even there are IBRO for the brain diseases. So you can apply a good project to get the seed grant. So that will help you to acquire some of the equipments that are needed in your lab to perform all these, at least basic level of genetic testing, like single gene test, or rather than go for the gene panel, you can start with a single gene set. You doesn't need a lot of equipment in that. So this is the point number 3 that you should actually do for that. And then you can also check out, for example, since this is a very growing field, so WHO has like a lot of projects running with the government level. So you can check with the WHO if your country's government has any kind of collaborative project going on. Then you can write to that project part and then ask like, okay, I want to contribute, my expertise is this, I want to be part of this. So this is another way. If you check with the WHO funded projects with your country, because WHO is funding a lot of projects in the field actually, but it is directly at the government level. So you have to check in your country if there is any option for such kind of thing. So this is basically 3 or 4 points so that you can look at. And regarding your points, if there is any commercially available kits for checking, yes, there are so many, but again, it depends on budget actually. So if you want to do basically a basic research, as I said, these single cell kits are not that expensive that you can get it. So if you want to do the whole genome sequence, yes, there are so many kits available. So even for example, there are kits which are very less expensive if you want to detect DNA sequence just for the swab actually. So there are it's a non-invasive technique. Commercially available kits are there with the buccal swab. You can find out the DNA sequences actually. Likewise, there is another company.
Thank you. The ones that you mentioned are more concerned with research. I was more concerned with clinical implementation, something that to get us started rather than because we have a lot of. I'm from Jordan, so and we are middle income country. So basically, we don't get a lot of funding. Most of the funding goes to the lower income countries, and for some reason, people think Jordan is rich, but it's not. Basically, it is considered as middle income country. And I'm sorry, but the last thing that you said something about that AI systems, AI tool, CEPIC guidelines, and the PharmJKB. Can you elaborate more on what exactly do you mean that these are AI tools?
Yeah, these are the AI tools. Let me first complete your second question part. So there's like, for example, if you see the Byron genetics, it can perform you for the all with the patient data actually. If you have a patient, you can just even if you don't want to do, just send the sample only. They can perform the this part. This is called the Byron genetics. So if you want to do a directly patient care stuff, so this you can do. Secondly, basically, there are like, for example, if you want to do anything which is like directly with the patient, so you can go with the Genomind. This is another company. You can go with the Sonic Genetics if you want to perform, for example, the inborn error of diseases like phenylketonuria, alkaptonuria, tyrosinosis, Klinefelter syndrome. So all these actually inborn error is actually basically one of the especially the Sonic Genetics. You can contact the Sonic Genetics. They can provide you with the kits. Likewise, if you are working with anything which is with the brain, so there is a Genomind that can give you the kits for that. So these are the some of the company I said, like the Byron Genetics in journal, or like Sonic Genetics for inborn error of diseases, and then Genomind if you want to work with the brain diseases. Yeah. So these are actually generally, if I tell you very precisely, there are 140 up till now. There are 145 kits available in all by the different companies for the disease actually. So there are, you can study actually various diseases, for example, in the pain management, in the mental health. So there are 145 kits which are exclusively focusing specific drug and these condition actually. So 145 are available as far as my knowledge is. Yeah.
Thank you.
You're welcome.
Fantastic. Okay, we'll take another question now.
Dr.
Okay.
Professor Liaquat Rada, you are audible?
Yes, Assalamualaikum, sir. How are you?
Welcome.
Long time.
Alhamdulillah, Alhamdulillah. You discuss all the aspects of pharmacogenomics, and it is very informative for us in Pakistan. And as you know that Pharmacy Graduate Association of Pakistan is working with the our team for the betterment of pharmacy profession since 15 years ago. So,
Yes.
as you know, we are NGO, and in this regard, we have a program for training for the pharmacy students
Right.
regarding pharmacogenomics, technology and its treatment, and other areas for the genetic therapy.
Mhm.
So my question is how we can manage these important areas like training session, and also induct our pharmacists in gene therapy system like AI and lab techniques. So it is a very difficult task, but we will inshallah do these things. On in this regard, you always help us in past. So how we can start this large scale therapy? The main question which highlighted by Madam from Jordan. Similarly, we have also these types of problems in our country, in Pakistan. So, suggest regarding the healthcare system through pharmacogenomic technology.
Okay. Thank you. Thank you for your question. So, as I know, I know about Pakistan, so I know the real dynamics happening there. So the first thing that can be done, because we don't have a very research culture there, and since this is one of the expensive techniques that need a lot of investment in that. So as far as also like we have like couple of good centers and hospital who are working with this. For example, HEGA in Karachi and Aga Khan Hospital in Karachi, Shaukat Khanam in Lahore. They are working on these themes to develop specific, precise, personalized medicine and the genomic studies. There is a whole center there, which is the lab of pharmacogenomics in Karachi as well. So what the PGA can do is basically align with them, as I said, number one. Number two is basically, since it's a pharmaceutical, it's related with the pharmacy, so like write a small project with the pharmaceutical companies and to fund them. Because otherwise, as an NGO without any funding, so you cannot proceed. So first of all is to make liaison with such centers who are working on pharmacogenomics and a pharma industry. So all together, like as I said, like this is a collaborative work. So without funding and without resources, you cannot proceed. So in order to get the fund, the way is basically to take the pharmaceutical companies in a line with that of those centers, like as I said, like some of the centers are there. If you there are also some people who are working at a very small scale in the university with the low budgets, you can also contact with them, so that can bridge something all together, and the thing can move forward actually. So this is the way to start there. Otherwise, because at the government level, not enough funding is there for research. So that is one of the key hindrance in moving forward there, to especially the field I'm talking about is something which needs some equipments as well as expertise. Beside this, need also money to carry on these experiments. There are so many diseases actually, so you have to perform each diseases, each drug, and then see if it is working or not. So it needs a lot of investment in using of the kits. So unless you are, maybe if the pharmaceutical company can sponsor 50 kits, and the center which is running this pharmacogenomic can provide you the resource, this can be the smallest startup joint venture can move forward actually. This is the way actually that seems a little bit hopeful if we move in this direction.
Right. Your guideline is very important and useful for us. But how we can reduce the cost of genetic therapy?
Okay, this is a different question actually.
Hmm.
The gene therapy is another thing.
Yeah.
Gene therapy is something different actually. So it's a next phase actually.
Yeah.
So we are not talking about like application of the genomics in you can say the finding out the variation of the changes into the gene level with the effect of the drugs. So this is the step one. When we talk about the gene therapy, that means, as I touched, like with the using of the CRISPR technique, we have to for example, utilizing different viral vectors, different plasmid, different promoters that can go specifically into the gene side and can express the new gene or suppress the any gene. So that, for example, let's suppose we know thalassemia is a genetic disorder, and there is a mutation in the alpha. If you talk about the beta thalassemia, which is a major thalassemia, so there is a change in the sequence of amino acid number 6. So if we can reach out at that level, and then we can replace that abnormal amino acid number 6 with the normal one, so then we can treat it. So we can treat it at which level? Because we know this is also a defect which is technically a genetic defect that is transmit from mother to a offspring. So that means there are two things. The person who's already born, we have to actually treat them. This is a strategy, but the treatment at the level of when it is inside the womb is different. So this is actually it's not that simple. We have to actually, there are so many technicalities, but yes, there are inborn error of disease which are treated at the level of when there is a developing a person inside the womb. So we are hopeful we can actually treat a lot of disease in the coming years. So, but it's not that easy. It's not that simple, because if I can tell to treat the epilepsy through the gene therapy, people are working since last 25 years. Still, like we can say, we just reached into the phase 2 of the clinical trial. So there is a lot of things that need to be considered before applying it to the patient. So you can imagine, like I myself work on like expressing the gene into the brain and then treat the epilepsy. So it's not that simple to apply the gene therapy directly. It might take a lot of efforts and technicalities, and then it need maybe 20 years or 30 years to treat some of the major diseases in the gene therapy. So gene therapy will actually be the next step. The pharmacogenomics will provide, you can say, the stepping stone to come up with the right gene therapy actually. So Baba Hamza, yes, sir, is also the hand raised.
Yeah.
Thank you very much, Dr. Professor Liaquat, for that informative lecture to us. My question is in the same context as Professor Nancy is asking. See, for the low-income countries, the full-fledged application of the pharmacogenomics is very difficult. Now, in the last five sessions of this, I have noticed that 4 or 5 diseases like you said epilepsy, psychiatric diseases, heart diseases, and the other day the speaker was talking about the single dose of cancer drug. So I think in the lower income countries, we can start with 2 or 3 major diseases where the mortality is more due to the wrong drug or due to DD reactions. So I think full-fledged application of the pharmacogenomics is very difficult in lower income countries, but we can start with 2 or 3 major conditions. I would like your opinion on this one.
Of course, I mean, this is the right way. Actually, the first thing is basically the first part always, whether it's any low-income country or any developing country. Actually, the major role is basically itself the government. Actually, government has to take this initiative. Otherwise, the second, the responsibility goes onto the pharma sector. The pharma company has to basically invest in this aspect, so that they can like come up with the, if you see the budget of the marketing of the pharmaceutical company is so huge. So just they should cut down the budget, and then they can like invest some of the part into the not huge amount, but like at least those labs who are working on these genetics is phenomena, so they can actually fund them so that they can start flashing the initial pharmacogenomic the basic infrastructure of the pharmacogenomic labs. So this is the point number 1. The point number 2, yes, of course, we cannot work all together in all the diseases. We have to specify which is the most common prevailing one, so that when you're going to develop a therapy, it can benefit most of the people. So each country has to look at which actually disease is more prevalent there, that can be better actually treated utilizing the concept of the pharmacogenomic and gene therapy. Yes, I totally agreed. You have to actually pinpoint 1 or 2 major disease burden of that country or of that region, and then start with that. Otherwise, we have so many diseases, we have so many drugs, we cannot focus all of them. We have to focus 1 or 2 major ones, and then see how it is progressing in that country and how much logistics and support it is there. And then if they are successful, and then this is actually starting. Yeah.
Thank you. Thank you very much. Nice answer.
Okay. Thank you so much, Prof. I see a question in the chat box by Adamu from Nigeria. It says, I'm currently working on an in vivo and in silico on diabetic study for my master's degree. Which aspects of pharmacogenetics can I explore to enhance my study, specifically using bioinformatics tools? And please, can you assist us with the slide after the class? Thank you. That's from Adamu.
Oh yes, I mean it's a huge, when we talk about the diabetes. For example, let's give you an example. For example, nowadays a very commonly one class is basically SGLT2 inhibitors or DP4 inhibitors or GLP-1 agonists. These are the common 3 class of anti-diabetic drug which is commonly used. So what you can do if you want to analyze the genetic variation of these drugs which are metabolized by which of the core enzyme. So this is one of the part that can be cheapened, that is to find out the genetic variants analysis of these CYP enzymes which are metabolizing these 3 class of drug. Number 1. Number 2 basically we have actually the tool available in which we have, for example, the genome-wide sequencing data, which is already from the different global studies, which is pull up there. So, but again, you have to actually utilize the local data to compare with the global data. So I'm sure if you are like working at the master level, you cannot perform these things that you can collect the pharmacogenomic data of your local people, and then you can compare it with the global available data, and then find out any specific variation which is found in that population. So one thing I said, like you can compare like SGLT2 inhibitors, DP4 inhibitors, and GLP-1 agonists, the variation and the metabolizing enzyme. Number 1. Number 2, basically what you can do at this level, you can just check the pharmacodynamic or the pharmacokinetic variation for example, of the bigonides, and because there is huge data available on this aspect of the bigonides and sulfonylureas. So what you can do is you see the impact of any genetic polymorphism on the pharmacokinetics and pharmacodynamics of the antidiabetic drugs, particularly the bigonides and sulfonylureas, because these are the very commonly studied one in the other countries. So you can see if any genetic polymorphism is exists that you can do in your local people there. Third aspect is you can do like, but you have to basically learn the AI actually. So if you learn the basic of the AI, what you can do, you can find out the biomarker actually. So there are integrated bioinformatics tool. If you know the little bit with the AI, you can find out the novel biomarker of for example, early detection of the diabetes 2 or the progression of the diabetes 2 in your population. So these are the things. For example, there are machine learning technique actually. So you can, if you have the data available of the gene which are responsible for the diabetes, for example, STK117A is one of the culprit gene in the diabetic drug response. There is another gene which called CCT5. These two genes are basically responsible for manipulating the drug response of the anti-diabetic drugs. So what you can do utilizing the machine learning tools, for example, SVM, the machine vector, and then you can do the GEEG. This is again the genomic variation, GW variations that you can find out. But again, you need to learn these technique actually. If you cannot learn this technique, you cannot proceed. AI is not just like, it's not chat GPT. Okay, you press up, you type a prompt and then you get an answer. No, you have to learn AI techniques, AI tools, machine learning tools, and for that you should have a good programming skill, like learn either Python, learn either R language, and you should know a good concept of the basic mathematics and statistics, so that you can apply these tools effectively and come up with, for example, the biomarkers that in your country or in your population actually. So these are the things that you can do basically actually. But again, you need to equip yourself with these learning methods of AI and machine learning. Otherwise, you cannot proceed without these things.
Okay, thank you so much, Professor. We're just on time now. I don't know if we'll still be able to take any question. It's we're running out of schedule now. I don't know. I think I still see one hand. Prof,
One only question, because I have another meeting in next couple of
exactly.
minutes.
Okay, please, Nagarajan, I'm sorry if I mispronounced, please unmute and ask your question.
Yes, sir. Hello, sir. My name is Gokul. I'm a PharmD student actually. So my question is very simple. As per the latest news and guidelines for beta thalassemia, CRISPR technology with the medical name called the Cas9 process has been approved by the FDA for treatment for the beta thalassemia patients. But due to its factor of being the highest cost of like around 2.8 million, what do you
Yes.
think, sir? Like based on in case if the number of patients are going to be increased, as not only beta thalassemia, I'm just open to any kind of disease condition. In case if the number of patients who are going to get into the genetic treatments increase, will there be any chances of cutting down cost, or will there be any kind of advantages in relation to that, sir?
Of course, actually, the therapy you are talking about, it was actually approved by December 2023, actually, and this is basically
Yes.
the examgorgolin autosetamel. Basically, it's a CRISPR based. It's not only for beta thalassemia, but like it was tested for all kind of sickle cell diseases, and actually the FDA only approved it for the beta thalassemia. But let me tell you, like, it is not only exclusive for beta thalassemia, but because as FDA only approved for beta thalassemia, but it has implication in all the sickle cell diseases. So you can treat all the sickle cell diseases utilizing this one, but which is not approved now, but like it's still, if you talk about the basic thing, so there is a BCL11A allele which is actually responsible for that. So they have actually they convert basically the fetal hemoglobin into adult hemoglobin. So this is a responsible gene is basically BCL11A. So with this CRISPR technique, they can simply treat it by the bone marrow stem cell and then convert the fetal hemoglobin into the normal hemoglobin actually. So yes, the answer is very simple. Actually, just to add on further to this, not only the FDA, but in January, the NHS, like in UK, is also for they actually not only approve it for thalassemia, but also for blood disorder, including the thalassemia and other sickle cell disease. So this is for the information. It's not only approved, but also like in NHS has also endorsed it and approved it in January 2025 to use that. Yes, of course, it's very costly. And again, the answer is simple. Yes, if it is like more common and the people are starting like utilizing this therapy more frequently, then of course as far as other therapies, it will cut down. Imagine when the mobile phone came in 90s, the cost and the size was too high, too big, and now you have like the
Yes.
mobile phone in your than a palm. So same is goes to, yes, of course, we as I said, like when the genome sequence was launched, so it was costing like $40,000. Now it's with you can do it like in a $200. So this is the way with this, but again, it will take time to get the cost reduced. Actually, like, the problem is that actually it's not something like a pharma drug that we manufacture a drug in a for example, in one batch you can produce 10,000 tablets. It's not like this, in which we have to take is patient specific, all the protocol, the things that we do is purely patient specific. The cord blood, either the stem cell that we are taking from the bone marrow, we have to process and refine and then put all this environmental and conditioning is actually patient specific. That's why it's not something that we can get and buy for and use it. No, it will be actually patient specific. So that is why it is very costly, because you have to do anything which is patient related only once. So this is the problem actually with this.
Sir,
At
is
this phase.
pharmacogenotherapy really possible for all the patients, irrespective of any other health conditions or sufferings which they're undergoing? So, say if a patient with beta thalassemia is going to get treated with gene therapy, in case of having any other comorbid conditions like say diabetes or hypertension, will it be really supportive and helpful for them to cure it?
This is a general clinical medicine question. If the patient is suffering, let's take an example of, for example, the liver transplantation. So all the patients are like who are because we recommend the liver transplantation when there is a 70% liver cirrhosis. Does all the patients are candidate for the liver transplantation having more than 75% cirrhosis? No. There is a score. On the basis of a score, we decide whether we should go for the transplantation or not. Same goes here. Not all the thalassemia patient will. For example, if the patient is very, very immunocompromised, we will never try that, because we have to put the patient in the immunocompromised medicines. When we put the patient in the immunocompromised patient, the immune system is very weak, so he will suffer from a severe infection. He will die because you have to put them for a couple of months on the conditioning regimen. So if you put them on a patient without any immune system, and he will be prone to diseases of infections, and he can die before the treatment even. This is the thing, that not all the people are candidate for that. There is a specific guideline and criteria on which we apply these therapy. Not all patients are candidate for that.
Okay, sir. Thank you, sir. Thank you so much.
Okay, thank you so much, Professor. It's
Okay.
been amazing. We're going to have to let you go now before we take some few announcements about the next session.
Okay, thank
so
you.
much, Professor. It's been
Thank you very
amazing.
much. Thank you.
From
Thank you, sir.
from
And
PJ, we are
Professor, okay, I hope
Yes.
it will be fine if I like sum up some of the questions, because I still see some questions in the chat box now that time will not permit us to take. I'll just compile them and send to you later. I hope that will be kind, sir. Would you appreciate that?
thank you, Dr. Professor Liaquat Rada
Okay.
from PJ.
Okay, thank you. Thank you so much, sir. My pleasure. My pleasure. Inshallah, hope to see you soon.
Inshallah, inshallah.
Inshallah. Inshallah. Thank you.
All
Thank you.
right.
Bye-bye.
Thank you so much, Prof.
Okay.
So right now, I'm going to tell us some things about the next session we're going to be having. because it's going to be an unusual session. Please just hold on for some time because you're going to have to get some information that will be useful for that session. So the next session we're going to be having is going to be the practical session, the hands-on session we're going to be having. So for that session, if you're using your phone, you may not be able to get the full benefit, the full experience as you're supposed to get. So it would be better to make plans to maybe get a laptop or borrow a laptop to ensure that you are able to flow along with the topic, with the speaker on that day. Because the way the session is being planned is such that we're going to see how to calculate actually pharmacogenomics. We're going to see how to implement pharmacogenomics at the point of care into practice. So it will be very nice that you have your computer with you on that day and you have enough sub as well, because we're going to be going to various websites to get data, CPEC, from GKB and the rest of them. So you want to be sure to ensure you have all of those things sorted out on that day. And that session is happening on the 28th. So you still have a couple of days before that time to make all of these preparations. And we are very certain that that session is going to be an amazing one. And as regards the session we're supposed to have, that's the day 4, the session we're supposed to have on pharmacogenomics in pediatrics care. We are currently making effort to still reach out to our speaker. That has not been successful, but however, we are not relenting. We are reaching out to potential speakers, other potential speakers who would be able to fill in that gap so that we also have that topic discussed with each and every one of us. So the details about that will be communicated by the next session about the schedule, the date, the time, and all. So we're going to call it a wrap now. Thank you so much for joining today, and thank you for staying back to listen to the announcement.
Thank you.
Thank you.
Thank you.
Sir. Thank you so much,
Yes,
Dr.
thank you
Zain.
very much.
Thank
Please, I have a
you.
question though. I don't know if
Yeah, please.
can I quickly
you can
ask.
it.
Okay. Can I get a recorded video of today's section? I kind of I came in late though. I think that was some
Yeah.
minutes to 5 Nigerian time.
Absolutely. The recording will be
Okay.
shared on the group as usual. Recording.
Okay. And if possible, to the one of yesterday as well. The session of the
Yeah.
yesterday too.
The one of yesterday
I
has been
appreciate
shared already on the group.
Okay. The video. That's the video record.
Exactly. It has been shared on the WhatsApp group.
Okay. I'll check it out. Check it out.
All right.
I appreciate it.
All right. Okay. Thank you so much, everyone. Bye-bye.
Bye.
Bye.
Thank you so much, Dr. Zain.
Bye.
All
Thank
right.
Thank
you.
you.
Yeah.
Thank you. Thank you very much.
Yeah. Bye.